Um, there's the recording, and I'm going to just introduce you based upon the flyer, then I'll hand it over to you. So our speaker today, Dr. Nivedita, supports the development of global applications on mass techs, AP MALDI, for tissue imaging and food-related contexts. She has a master's in microbiology from the University of Calcutta. She has forayed in the field of MALDI mass spectrometry during a PhD from CSIR National Chemical Laboratory in Pune. During her doctoral stint, she developed high throughput methods for clinical and microbial metabolomics. Gosh, I know that word, metabolomics. She followed it up with her postdoctoral studies at Boston University School of Medicine. So that's quite cool. With MALDI, mass spectrometry, imaging on the brains of athletes suffering from chronic traumatic encephalopathy. Wow. She develops mass spectrometry. She believes mass spectrometry is one of the cutting edge technologies that can prove to be the most precious workhorses for biochemists. So in a talk, she's going to introduce the audience AP MALDI and share some of the recent applications of mass spectrometry imaging. So with that, I'm going to hand it over to our speaker. Take it away. Thank you.
For a wonderful introduction, and thanks again, everyone, for inviting me. So let me share my screen. Just are you guys able to see my screen?
Indeed, yes.
Presentation mode. Just let me know if you can.
It's all good.
All right. Today, tissue imaging. Okay. And I must tell at this stage that, Mastech is the company that produces AP MALDI ionization sources or atmospheric pressure MALDI source. And it's the distributor, it's the local distributor in South Africa for Mastech products. Okay. And of course, like as I mentioned before, thank you so much, Metabolomics South Africa, NSA, for inviting me and giving me this opportunity to talk. So, you can talk with, Mark from Anatech and Vishal is from Mastech. So at any stage, if you have any questions about our products, of course, I'll also be there. Okay. So what is, MALDI? I have based this presentation from a very introductory perspective, and that's why feel free to ask any questions. I would rather want it to be more interactive as journal clubs are. And if you have any questions, please feel free to interrupt. Okay. So I will start off with MALDI. Okay. So what is MALDI? As you can see, this is a plate. Okay, where you spot your sample with a matrix. Now, in this, what is a matrix? A matrix is a small molecule compound, okay? And that compound is important for absorbing UV light and transferring that to a small molecule or any kind of molecule, say a metabolite or a lipid, to be ionized. Okay, so the ionization happens because matrix absorbs the laser energy, which is this thing. That is, the laser is hit on the matrix and analyte, and then the ions are formed, and they are, important inside the mass spectrometer. Now, the important thing to consider is there are different kind of MALDI sources. Okay, you can have a traditional MALDI, which is operates under vacuum, and then you have a MALDI which operates under atmospheric pressure or in ambient condition, and that is what we do or we produce or we manufacture. So this is the small footprint atmospheric pressure MALDI source that we manufacture. And this is something that can help a lot in tissue imaging. Now, I talk a little bit about MALDI mass spectrometry imaging, just introduce the technique once, and then talk more about the product. Okay? So what is MALDI mass spectrometry imaging? So this is a method which is complementary with routine histopathology techniques such as HME staining. There are different other staining procedures too. You can identify and localize various biomolecules on tissue based on their M/Z ratios. Okay? It does not require any cumbersome labeling or expensive antibodies, which traditionally for any kind of antibody staining you would require. Okay? It has been applied to various research areas for pathology, diagnostics, metabolic pathway analysis, and for plant research too. Now, this is a conventional workflow for MALDI mass spectrometry imaging or AP-MALDI agilments. Okay, the first thing that you do is you harvest an organ. Okay, it can be an animal tissue. It is also the same process is applied to plant tissue as well. Then what you do, you section the tissue, and after that you deposit matrix on it. Then you do a mass analysis, and finally you do data processing or image reconstruction. For our audience today, I would be mostly focusing on AP-MALDI and the various applications that you can do and might be helpful for you. Now, I'll show you a small video. Let me know if you can hear the video as well. Although, there is no audio as such, but this is something that I would like you to see. So here you would see how easy it is to start from. So say if you have a system such as an Exploris, okay, an Orbitra, you remove the ESI source, okay, you put a small capillary extender that is required for and transfer. And finally, the AP-MALDI source in, you fit it in with the flanges and plug in some of the cables to start the operation. And that's it. It is as simple as that. So if you have any LC-MS system, you just remove the ESI housing, you plug in our source, and then the system becomes it's fit for to do any tissue imaging. Yeah. Now, going to the next slide. This is how AP-MALDI looks like with different Orbitra platforms. Okay. As you can see, we are completely compatible with different generations of Orbitra, like with Orbitrap Exploris, like 120, 240, 480, we are completely compatible. With the Tribrid series 2, we are compatible, and with the Q Exactives as well. And in fact, with LTQ Veloz also, we are compatible. In fact, we are also compatible with multiple OEMs, say some Q-Top and triple quads also, we are completely compatible. And I must tell you, if you have a matching capillary extender, then you can also take the same source. I mean, say if you have a triple quad with a matching capillary with say Exploris, then you would be able to convert that to an imaging platform. This is how the entire thing looks like. As I have already shown you that you fix it. So how do you what are the different components? This is the slot where that opens and you insert the plate. Okay. Then you have the instrument port which fits. You have already seen that. You have power supply, LAN cable, laser on indicator, and external control port. Okay? These are the different. Yeah. Any questions? Yeah.
Sorry, Mohammed, can you just mute your mic, please? Thank you.
Can you please repeat, Shane? I mean,
sorry, there was a mic open, it's been muted. Sorry about that.
Oh, okay, fine, all right. So these are the different specifications, okay? And the one of the most important thing is it is kind of very easy to carry, and it is it has very small footprint, as you already saw. Okay. And there are two different formats on which, how you can see it. That is, one is the high throughput analysis mode where you spot your samples and then you can just do quickly analysis of say some metabolomics extract. If you have, you insert the plate and you do the run very quickly. That is say one spot requires 1 minute or sometimes even less than that. Okay. And the other one is mandated for the tissue imaging where you put in your slide and then you scan across the slide for getting the data. Now, softwares that are recommended for and that operates AP-MALDI. Target NG software does AP-MALDI high throughput analysis and Target Next software does imaging. So these are the three different steps to get your tissue imaging data. It's very easy. You load the slide, you define your region of interest, and then finally you acquire your data with MS. Okay. These are the tissue imaging applications that I will be talking about. But before that, I just wanted to understand, does anybody have any questions till now?
If anyone has any questions, just throw your hand up. Usually we have questions at the end of the presentation. I'm sure there will be some questions.
Sure, no problem. Okay, so right now I'll be focusing a bit on the different tissue imaging applications. Okay, so I would talk about is spatial resolution. Okay, so you can go from as low as 10 micron to 100 micron. Okay. And I will be showing a few examples of the spatial resolution that you get with high resolution mass spec and with our system. Okay? Now, a combination of a high resolution mass spec and high ultra NG-UHR system is you acquire an arbitrage data, which anyways is high resolution. Now, if you want to look at localization of it, so these are three different lipids, okay, which are very close masses, as you can see. And all of them show very different distribution in ring. Okay. And this is the power of high resolution mass spectrometry based imaging. Okay. The same thing you can also do for some of this molecular imaging that you do for skin, where you see the kind of penetration of any drug or how how much of a drug has entered inside say skin or say some follicle. And the part is you can look at your compound of interest along with the say lipid or endogenous compound together with the different things with this AP-MALDI imaging. By the way, this has been done by our European collaborator lab, which is NIST, Luxembourg Institute of Science and Technology. I think some of you might have already interacted with Jill during the last workshop that happened in May, I guess. So I also wanted to talk a little bit about why atmospheric pressure MALDI becomes very important. So this is one example that I wanted to show. And this has been done in our long-term collaborators lab. So she was looking at conventional MALDI and she wanted to see the distribution of gangliosides in brain. Now, one of the things that she got, she got the distribution, but then she was not very happy with it because the antibody staining did not match with GM1's location with whatever she was trying to look for. So she realized that the compound or gangliosides were getting in-source decay or in-source were showing in-source fragmentation. And she did some signal normalization and that reduced the in-source decay. Okay? Now, what happened was when you do this in-source, if you are encountering in-source fragmentation, there is also ways to reduce this. So one of the ways is using some different matrix. And in this, she chose a matrix dihydroxyacetophenone. And what she observed was the in-source fragmentation definitely reduced for the gangliosides with conventional MALDI. But she faced another problem, that is since these runs can take sometimes hours, her matrix started sublimating inside the source, because this operates under vacuum. So then she realized that she probably needs to try out an atmospheric pressure or an ambient source to understand that how gangliosides look like. And she again, performed was whether the in-source decay is reduced. And she found that in-source decay is reduced with AP-MALDI when she was using the new matrix. The next point that she had was whether the matrix is subliming or not. And the matrix did not sublime when she was using AP-MALDI. Finally, she did discriminate a lot of isomers as well with this sticking. She also found that with AP-MALDI, very less amount of matrix is required for imaging a tissue sample compared to intermediate pressure MALDI. And about 25 times less matrix is required with AP-MALDI compared to intermediate pressure MALDI. This was one of the observations, the initial observations that she had, and it has been consistently seen by some of our other users too. So coming up to lipid imaging, this is one of the examples where Mass Spec source with Orbitrap Exploris 480 could get a lot of lipids data. Now, I'm showing here just a panel of 16 lipids, but trust me, this is not 16. There can be multiple analytes that you can get from one run. Okay, and the interesting thing about lipid imaging is that you can use different kind of matrices and have your own choice of which lipids you would be lipid analysis and lipid imaging with MALDI, of course, is one of the hottest fields, and you would be able to see a lot of lipids. Now, phosphatidylcholines, ethanolamines that are indeed done. I have also shown one example of ganglioside. I also want to show you this work where cholesterol, which generally is very important for brain functioning, but its identification and analysis sometimes becomes very difficult because cholesterol fragments very easily, and the detection, that's why, becomes very difficult. Okay, so in this, what they have done, they have used Gerard P derivatization. And after using Gerard P derivatization, they were able to see cholesterol and some phospholipids too. So this is one of the ways in which, if you would want to do some derivatization for say a specific class of molecules along with some say lipids, you can do it very well. Okay? It's easier with this way. Now coming back to the part which we all are very interested in, that is metabolites. Now, here I'm showing you the imaging of metabolites in kidney. This has been done by one of our user in UT Austin. You may have heard of her, Dr. Erin Seeley. And in this work, what you will see that she has shown different kind of comparison in which, as I mentioned before, that you require very less amount of matrix. And she has seen that in some cases, you can also get signals with 1 to 2 layers of matrix, which is incredible. Because that means that the ionization is also softer and you can get a lot of coverage for metabolites. And this she's showing for arginine amino acids and some spermidine and polyamines. Okay? I also wanted to show a few work in the plant imaging domain, which pertains to metabolites. And here, what you would see is there's a comparison of vacuum MALDI and AP-MALDI platforms for some of the metabolites that are involved in salt stress tolerance. Okay. So what you observe is these are control and salt treated nodules, and AP-MALDI is good enough to show some, even if you have salt. I mean, the sample prep is such that it can withstand a lot of presence of salt and any sort of salt interferences. And we were able to identify again, like, small molecule metabolites. And it was a very elegant study which can show if somebody has any plant metabolomics content to just how you can go from one stage to another. There are, and of course, these metabolites were showing very good classifier between the salt stress tolerance and like non-salt stress tolerance. I would also like to show you something that we did in India, where we did a plant breeding for strain improvement. This was done in collaboration with NRCG, and which is a agricultural research institute. And of course, like as you may also relate to in South Africa, that some of the challenges that face global south are probably a little bit different when it comes to food security, how the cultivars or like plant breeding can improve shelf life. That is very important in our contexts. So with that, this plain seedless variety was bred with Pusa Navrang to get a variety called as Manjari Medica. So this Pusa Navrang variety produces a lot of juice, but it is not very sturdy. So, it doesn't have a very good shelf life. Whereas plain seedless, as you may know, it has a very good shelf life. So when it was bred, the phenotypic characters showed that Manjari Medica has properties in which like the juice producing capacity is very good, but it has a longer shelf life too. So with this, we thought of collaborating with our, they are also long-term collaborators of ours, NRCG. So Vishal and I are part of this project, and we thought of, understanding how it looks like when we image a grape. Okay. I will be showing part of some of this data. The data is very complex. You can always we can, if any of you are interested in talking about it, we can talk about it later. So these are the three different varieties. FS is plain seedless, MM is Manjari Medica, and PN is Pusa Navrang. And here the distribution that you see, they are very interesting. Arginine is very, very important for shelf life. And you see that in flame seedless, it is in higher quantities, and that is how it should be. Whereas in Manjari, in Pusa Navrang, it is in less quantities. And in Manjari Medica, it is in middle, somewhere in the middle, which shows that it is inheriting the properties of like arginine from flame seedless. Similarly, if you look at proline, proline is a very important marker of stress. And what we observed was proline is again coming somewhere from flame seedless. That is like Manjari Medica is inheriting it from flame seedless. And this is a very important, one of the very important results for our work. Um, I will also show you one small example of like some leaf imaging. If somebody wants to do, you can look at various metabolites. You can just take the leaf, you can just remove the cuticular lipids, and then you select an area. I mean, here we have selected a part of this area, and this is the image. So here what you see is the total line chromatogram. That is, there can be multiple analytes which are present. And here I'm picking up only one analyte, which is adenosine. You can have a lot of data and a lot of metabolites that one can get. I also wanted to give you one example of lipids and neuropeptides. So when somebody has to do some peptide imaging, ideally the workflow is different, okay? Because lipids are such abundant molecules that they sometimes interfere with the distribution of peptides. You won't be able to see them. So here in one of the work, lipids are shown first of all in uh, a crab's brain, and then the neuropeptides are studied. What was interesting to note is these two workflows are different, but there is always a possibility of combining workflows in which you don't have to, do any sort of delipidation. And then you can also get like, kind of peptide information. How you do that? As I mentioned, that lipid detection with MSI involves, actually, it doesn't involve any washes, and peptides detection sometimes require a lot of delipidation. So if you can combine the workflows, it can save some time and reagents. One of our customers, I mean, you may again have heard of her, Professor Lingjin Li. She has actually developed some reagents which are click chemistry based, and these reagents bind with the peptides, okay? And you don't have to wash anything. You are not using any washes. So you would be able to see the lipids as well as the neuropeptides, okay? So in this case, what you see, like when they have like a control and treated, like that is the reagent, like, something derivatized with the reagent that they have developed, you will be able to see, the lipids and neuropeptides both in the same workflow, whereas in the control, you wouldn't be able to see that. I am showing another example, which is like, drugs or like any pharmaceutical compound, how you can see their distribution. So pharmaceutical compounds distribution and endogenous lipids is possible with AP-MALDI. Here, pure glitazone is used, and initially, you would see the distribution of pure glitazone, and in these are all endogenous lipids. Now, the important thing is when you are identifying with HRMS, you also have to confirm it, right? I mean, that whether it is pure glitazone or not. So you can do a, MSMS or tandem MS imaging of the same compound. And this is what, like, Professor Enrico De Volli did. They did a on-tissue based MSMS or tandem MS, and then they looked at the MSI distribution or the tissue imaging distribution of it. Once they had the confirmation, they wanted to see whether you can quantify it or not. And they were able to see quantitative AP-MALDI MSI of buglitazone at, the different hours. This was according to the ADME, kinetics that they were expecting. And that is something that they have found. So these are some of the applications that I wanted to talk about. I also wanted to show you, like, some of the other very important aspects of like any tissue imaging. One of the most important aspects is like, the matrix sprayer. We recommend, like, if you go with Sun Collect sprayer, you can actually do a lot of, different kind of analysis. Some of the analysis that I showed with grape and leaf, we have done it with Sun Collect. We have developed the methods with Sun Collect. And here what you see, in this, they have spotted different concentration of drugs in a treated and in an untreated liver tumor. And they wanted to see how the distribution of, like, that drug looks like. And we were able to quantify about 0.1 microgram per ml of, this drug that they wanted to see. And the same thing was again imaged in liver. So these are some of the examples that one can, do with matrix sprayer. And another very important thing, that is image reconstruction. That is like, finally, when you get your data, you have to do the data analysis and interpretation. So Mosaic is one of the softwares that is like, recommended for doing, your imaging data interpretation. There are other softwares too, like such as MSI Reader Skills and all these softwares, which, like, you can use for looking at how your data or where the molecules are localized. Some of the features of Mosaic are, it allows you to, like, add the histology data, or say if you have any kind of other co-registration formats that you would want to add, you can very well add that. And again, like, you can have some ways in which, calibration, and then overlaying of the data that is possible with this software. Finally, these are some of the pointers. This system is completely compatible with Orbitraps, almost with all generations of Orbitrap, that is LTQ, starting from LTQ to Explorers. We have capillary extenders which are available for all generations. Okay, so you can always have that option of if you have an Orbitrap, and you can always switch it to an imaging mode. Now the source switching, as I mentioned, it takes very less time. You don't really have to wait for a long time. Matrix choice, it is compatible with different matrices and solvents. Tissue imaging, as I showed you, that it is possible with excellent spatial resolution. Amount of matrix required, it's comparatively less than conventional vacuum MALDI. And quantitative HRMS is possible. I haven't dealt a lot with, at least for this presentation, I haven't shown you anything about anything other than tissue imaging, because that was the focus. But you can do a lot of applications if you say just want to take some tissue extract and you want to see, or say some biofluid extract and you want to see what metabolites you have, you can spot them and directly see if you can find anything. You also have compatibility with triple quads. These are the different matrix deposition options. Like one is of course sun collect, and you can also have sublimation and other things that if you would want to do that. This source is completely portable, and it comes with a Pelican case where you can just keep it. And finally, I would like to show you where all you can find us. So we are going to be present in one of the conferences in a workshop next week from July 28 to 30. Then at IMSC in Melbourne, we are going to be present. So we are having a workshop towards the end of August in India. This is the first mass spectrometry imaging workshop that we are going to have in Bangalore. And finally, we are going to be present at IMSIS conference, which is going to be in Münster, Germany. Okay, so yeah. So if you have any question, please feel free to ask me and we can just discuss now. So yeah, the floor is open for you to ask me questions. Thank you.
Thank you so much for such a wonderful presentation.
Thank you.
Are there any questions for the presenter? You can either put your hand up and unmute, or you can type in the chat box if you're not too comfortable speaking orally.
Yeah, I mean, we have all been there, so we know.
So while we wait for questions, I always ask the presenter if would they be willing to provide the email address for anyone who wants to follow up with questions or just chat about the technology or possible collaborations. I think in the chat box.
Yes, yes. Should I give it? I'll probably pass it to Shane. You're, I think, marked in the email, or Malati, you were there. So Mark anyways has my email ID. So you can always ask any questions if you may have. And I understand, sometimes when we are introducing a technology, it sometimes happens that the questions do not come at that point. So at later, if somebody has any questions, they can feel free to kind of ask me anything. So
okay, yeah, anyone who has questions later can contact me and I'll forward it to the presenter. Everyone should know my email at nmr.nw@gmail.com. It's also on YouTube for people who watch later. They might be interested and might have a question or two. So those that are watching later after this live presentation, you'll feel free to contact me with some questions and I'll forward them to the presenter. Out of interest, can you raise your hands for those that did the imaging workshop earlier this year in May.
Uh, okay, somebody has a question. Okay, let me first
And
answer the question. Felicity
okay.
has asked this question. Can you use frozen sections and paraffin embedded tissue sections? Okay, great question, Felicity. So yeah, frozen sections are recommended. So say if you're doing lipid or metabolite imaging, of course, go with frozen sections. So paraffin embedded sections are also done by one of our kind of collaborators and users, of course. Although I think you may have attended his talk or workshop in May, Peter Barhat. So he has a lot of expertise on FFP section. So it completely develops on depends as well on what applications you are interested in. So say if you would want to look at peptides, go for FFP. That would be fine. I don't think you would face any issues. But for metabolites, I think the first place to start would be frozen sections or fresh frozen sections. Great. Somebody asked me a question. I'm so happy. Thank you, Felicity. Yeah.
I
So,
know.
Shane, yeah,
One of my PhD students is currently doing proteomics and metabolomics of brain tissue. So
mhm,
once she's identified some important metabolites and proteins, we definitely want to go to the imaging route to try and detect these specific metabolites and proteins and see the distribution in the brain
sure.
in this because specifically it's an immune response we're looking at. So we want to see how it distributes with all these immune cells.
So of course, I mean, that would be a very good, of course, you would have the validation from proteomics and metabolomics to at least know which analytes or which metabolites and peptides you are interested in. So definitely, you can at that point of time go for tissue imaging and see kind of how it can be done. So yeah.
Yeah, so if anyone's thinking about how to apply this, that once you have that metabolomics or proteomics data, what's the next step? And imaging is a great next step to try and confirm, validate, support whatever you're saying relating to the metabolomics work that you did.
Yeah.
Yeah. So
Yeah.
while we're on that, what are some of the elements to be mindful of when you at the experimental design level, if you are about to go and do an imaging experiment? I'm just thinking about managing expectations.
That's a great question. So, one of the things that, kind of you have to be very, very considerate about when you are starting off is tissue imaging's sample prep. So, right from, kind of collection, that is harvesting of tissue, to your, sectioning as well, you have to be very careful. So, there are, some sectioning protocols which are recommended for tissue imaging. They are widely available, in literature, and that is how, that is the first step when you have to, at least know that, uh, kind of how you're going to proceed with the sample prep. Now, the next thing that is also important is the context. So, for example, Shane mentioned that he probably will go to the tissue imaging route after he is done with the proteomics and metabolomics, and he at least understands which he would want to look at. So that is something that which is very much possible for anybody to first define your context and then see what you would want to get out of it. Although with HRMS you would be able to get a lot of kind of data, so that definitely is possible, that sometimes whatever you are getting out of tissue imaging, that can complement your metabolomics and proteomics data too, because you would be getting not the bulk effects but only the tissue level effects. So yeah. Okay. Molagon has a question. Sorry if I mispronounced it. Okay. For frozen section, since OCT is not recommended, great. Yeah, is it possible to use conductive double-sided tapes to mount the sample? Okay. Now, the most important thing to remember here is, of course, OCT is not recommended. There are other mounting medias. Okay, so for example, somebody can use carboxymethyl cellulose or gelatin to mount or like to prepare your sample and then mount them on the slide. Okay, and excuse me. So double-sided tapes, like, kind of say if you are using some tissues such as leaf, in that case, it's fine. But I wouldn't recommend that for anything like animal tissue. That may not be, very good. Yeah. Does it answer your question, Mola Monyen? If there is anything else you would want me to add, I can add.
I think that covers the question. Um,
Fine.
are there any other questions for our speaker? She seems very eager to answer some questions.
Yeah, I'm very eager to answer the questions. So,
In the case of there was a paraffin embedded. So you also get formula fixed paraffin embedded. Yeah, those ones that have these very waxy exteriors. Do you have to remove all that wax in order to do the? Okay.
yeah, you would have to remove, that is deparaffinization you would have to do. And once you do the deparaffinization, after that, you would be, able to, kind of do some, I think there, you may have to do some dehydration. And after you do the dehydration, you spray the matrix and then do HMRD MSC. I think
Okay, so
Peter probably
it's
has
basically
more
the
about
same
it.
for metabolomics as well. We have to do the whole xylene wash, get rid of the wax, and then rehydrate it, and before we can actually start looking at it. So pretty much the same procedure then.
Yeah, yes, exactly. Okay.
Uh,
Okay,
And
Felicity.
what section thickness is best?
Yeah, Felicity is asking me a lot of questions. Felicity, may I see you? Okay, Felicity doesn't want to talk to me. She can just chat. She will just chat with me. Hi, Felicity.
Hi, how are you doing?
I'm great.
Yes, we are. Yeah, we are at TUT and we are starting a study in my PhD. And I'm actually working on plant material, specifically specific roots. And we've been having problems getting our sections fixed to the slide and getting our mass spec to give us results. So that's where me and Renat is asking a lot of questions.
Completely fine. So, yeah, since you mentioned that you are interested in root, okay, see, ideally we recommend a section thickness of about 10 to 15 micron, okay, but I have seen in some of my experiments that if we even go a little beyond, like, 20 micron, it's okay. So MALDI can handle like the section thickness. It's just that, yeah, I would not want the sample to be like, kind of very thick, because in that case, it may cause a lot of like, an interference and all. But ideally, if you can just like, kind of have a section which is say 10 to 15 would be the ideal, but you can also go for like 20 or 25. Just check it out, because this sometimes it so happens that you may have to optimize a bit to see which one works for it. In fact, there is a paper which recently came and it says that MALDI can in fact like go as thin as like 5 micron. And in some of my experiments I have seen that like I have so the grape data that I showed, I have actually sectioned it with a blade. So you can imagine, it's not microtomed, or like it is not cryomicrotomed. So yeah, that's kind of great. So yeah. Okay. Yeah. So both of you have the same question. I hope I have answered to it.
Yes, you've mentioned that you did freehand sections now. How did you now get the section to stick to the slide if you did the freehand section?
So, yeah, so I have done with this like, berry, right? I mean, grape is a berry. Now, the good part about grape is that, like, it has a lot of juice. So if you just like place the section, you have to take a very thin section, just keep it for some time and then desiccate. I mean, I desiccated it for at least half an hour so that there is no moisture that remains, but then the berry sticks to it, okay? Sticks to the surface. So that is something that I did and it helped. Now, you can, in the case of leaf, what I did was I used carbon tape to Vishal, are you there? Oh, Vishal, I think has left. Okay. So in the case of leaf, what I did was or rather what we did was we took the leaf and we fixed some carbon tape, and that is how we managed to keep it on the surface. In fact, there's a recent publication that came that uses roller tape to a double-sided roller tape, and on that they have placed the leaf, and they are able to see some very cool distribution of metabolites from the leaf surface. So yeah, if you guys want, I can forward the link of that paper so you can look at that.
Thank you. We will definitely get in contact with you. Thanks, Emils.
Sure. Thank you so much, Felicity.
Or at least we got one contact out of this, and it is a success, this journal club. So thank you so much for the presenter for sharing with us. Yeah. And
sure.
I will forward the email to you, and I think they have lots of other questions to discuss with you.
Sure. Yeah. Please feel free to get in touch with me and my colleague Vishal as well. In fact, we are doing some very cool plant related applications, and we would be happy to answer any questions that you may have or anything related to animal tissue too. So yeah. Okay.
Mark, you have anything to say? I see he unmuted for a moment. No. Okay. Okay. I think with that, we'll close today's journal clubs. Thank you so much to the presenter, and thank you for the discussion involved afterwards to our colleagues.
Sorry, Shane, I managed to unmute myself there. Sorry to interrupt. Just no, nothing further from our side. Be nice to actually chat with you, Felicity. Your account actually falls under me at TUT, so I can schedule a meeting with you at some point.
I'll include you in the correspondence, Mark.
That'll be great, Shane. Thank you very much. Yeah, pleasure, Felicity.
Okay, great. We have a connection made.
Yes.
Awesome. Okay, thanks everyone for attending, and thanks to the presenter. It was a great journal club.
Thank you all. All right. Have a very good rest